Welcome to G Fun Facts Online. Today, we're diving deep into a silent crisis happening in our oceans and, surprisingly, on our dinner plates. Sharks, the apex predators that have patrolled our seas for 400 million years, are vanishing. It's estimated that up to 100 million sharks are killed every single year. And while the brutal practice of shark finning gets a lot of attention, a shadowier, more pervasive threat is the global trade in shark meat. This multi-billion-dollar enterprise thrives on deception, but scientists are now fighting back, using DNA detective work to unmask a global chain of illegal activity. The statistics are staggering. Since 1970, global shark and ray populations have plummeted by a catastrophic 71%, mostly due to overfishing. The shark meat trade has now surpassed the fin market in both volume and value, estimated at $2.6 billion between 2012 and 2019. What makes this trade so insidious is mislabeling. A recent, shocking study found that an astounding 93% of shark meat products sold in the United States were either mislabeled or so vaguely described that consumers had no idea what they were eating. Fillets are often marked with generic terms like flake, rock salmon, or just shark. Using DNA analysis, the study found that 31% of the products tested were from endangered or critically endangered species, including the great hammerhead. These highly protected species were being sold for as little as a few dollars a pound. This means well-intentioned consumers are unwittingly funding the extinction of some of the ocean's most threatened animals. This isn't just a tragedy for biodiversity. Sharks are keystone species. They regulate the populations below them, maintaining the delicate balance of entire marine ecosystems. Removing them can cause a chain reaction, leading to the degradation of coral reefs and seagrass beds. What's more, large sharks accumulate high levels of toxins like mercury. When their meat is mislabeled and sold, it poses a significant public health risk. The core of the problem for law enforcement is identification. Once a shark is processed, its head, fins, and skin removed, it's nearly impossible to tell what species it is. This is where the power of DNA comes in, providing an unassailable tool to peel back the layers of deception. In the world of wildlife forensics, DNA is the gold standard. Several powerful techniques are now at the forefront of this battle. The most fundamental is called DNA barcoding. Think of it like a universal product code for every species. Scientists use a short, standardized segment of an organism's DNA. For sharks, it's a part of a gene called COI to identify it. The process is remarkably precise. They extract DNA from a sample, whether it's a fresh fillet, a dried fin, or even pet food. They amplify that COI gene and sequence its genetic code. Then, they compare that code against a massive public reference database, like the Barcode of Life data system. A match provides a definitive species identification. DNA barcoding has been used all over the world to expose the truth. In Singapore, a major hub for the shark fin trade, researchers analyzed over 500 fin samples and found 27 different species, 17 of which were listed as threatened. A study in Brazil tested 427 samples from local markets and found that a shocking 53% were from species threatened under Brazilian or international law. The evidence is clear. Our marketplaces are flooded with endangered shark products, hidden in plain sight. But what happens when the products are heavily processed, like in shark fin soup or dried jerky? The harsh processing can degrade the DNA, breaking it into small fragments. This makes it difficult to read the full-length barcode. To solve this, scientists developed a technique called mini-barcoding. Instead of the full 650 base pair barcode, this method targets a much shorter, more stable fragment of the same gene, typically between 100 and 200 base pairs. These mini barcodes are more likely to survive the processing, allowing for successful identification. One study showed mini-barcoding could reliably identify processed fins from 7 out of 8 CITES-listed shark species at the time. It even successfully identified the species in over 60% of shark fin soup samples. This technique has been a game-changer for tackling the most challenging products. One of the most innovative frontiers is the use of environmental DNA, or eDNA. All organisms, including sharks, constantly shed genetic material into their environment through skin cells, mucus, and waste. Scientists can now collect water samples from the ocean, filter out this trace DNA, and identify which species have recently been in that area. This is a powerful, non-invasive tool for monitoring shark populations, especially rare and elusive species. It helps scientists understand where sharks live, feed, and breed, providing crucial data to design effective marine protected areas. For instance, eDNA has been used to detect the presence of critically endangered great white sharks in the Mediterranean, a region where they are rarely seen, allowing for targeted conservation efforts. And finally, to take it a step further, scientists are using population genomics. This goes beyond just identifying the species. By analyzing thousands of genetic markers, researchers can identify distinct populations of the same shark species from different parts of the world. This is a game changer for law enforcement. With a genetic reference map, they can analyze a seized fin and determine not just that it's from a protected species, but also which specific region of the world it was likely poached from. This helps pinpoint illegal fishing hotspots and dismantle international trafficking networks. Together, these DNA tools form a powerful forensic arsenal, shining a harsh light on the dark corners of the illegal shark trade. But this science is only as good as its application in the real world of law enforcement and justice. Fortunately, genetic evidence is increasingly being used to build robust legal cases, leading to successful prosecutions and seizures. In Australia, a landmark case involved a large seizure of illegally held shark fins. The Australian Museum's DNA lab was called in. Using their techniques, they identified fins from numerous Australian shark species, including some listed as vulnerable. For one seizure, DNA analysis revealed the fins belonged to no fewer than 22 individual sharks. This concrete evidence was instrumental in the prosecution, leading to convictions and fines totaling over $23,000, a powerful deterrent. In Brazil, DNA analysis of 3 separate illegal shark fin seizures identified at least 20 different species from 747 fins. Many were endangered and protected under Brazilian law, providing undeniable proof of environmental crimes. A major challenge for customs officials is speed. They often have to process huge shipments in under 24 hours. Traditional DNA analysis, taking days or weeks, is often too slow. To address this, scientists have developed rapid, field-based DNA tests. These import tools can provide results in a matter of hours. For example, a new real-time PCR protocol can screen for 9 of the most commonly traded protected shark species in a single reaction in less than 4 hours for less than $1 per sample. A positive result gives officials the probable cause they need to detain a shipment for more detailed forensic analysis, which can then be used in court. These kits have been successfully implemented in Hong Kong and Colombia, empowering customs officials to clamp down on the illegal trade in real time. The primary international agreement governing this trade is CITES, the Convention on International Trade in Endangered Species. It provides a legal framework to ensure that trade doesn't threaten the survival of wild animals. Species are listed in appendices that afford different levels of protection. Appendix 2 is key for sharks. It includes species that may become threatened unless trade is closely controlled. To trade an Appendix 2 species, you need a CITES permit, which requires proof that the trade is sustainable and the specimen was legally acquired. In a monumental decision in 2023, the entire family of requiem sharks, that's about 60 species, including tiger, bull, and blue sharks, was added to Appendix 2. This, along with previous listings for hammerheads and makos, means a huge portion of the international shark trade is now regulated. But these listings are only effective with robust enforcement. And that's where DNA becomes absolutely critical. It provides the scientific proof needed for customs officials to verify that a shipment of so-called legal shark fins doesn't contain protected species being smuggled within. While DNA analysis is a revolutionary tool, its path to a courtroom conviction is not without hurdles. One of the biggest is DNA degradation. Fins that are dried, bleached, or boiled can have their DNA broken down into unusable fragments. Even with mini-barcoding, some samples are just too degraded to yield a result. Another major challenge is the reference database. A DNA sequence is meaningless without an accurate, comprehensive library to compare it against. While these databases are growing, they are far from complete, especially for rare and cryptic shark species. Building these libraries requires a massive global effort. Then there's the cost. While sequencing has gotten cheaper, running a DNA forensics lab is still expensive. The equipment, reagents, and trained personnel are a significant barrier, especially for developing nations that are often on the front lines of the illegal trade. Finally, there are the legal hurdles. For DNA evidence to be admissible in court, prosecutors must prove an unbroken chain of custody for the sample. They must show the scientific method is valid, the lab meets quality standards, and the results must be presented by a qualified expert. The entire process has to be forensically sound. Despite these challenges, the future of this fight is bright, thanks to an evolving arsenal of DNA tools. The rise of portable real-time DNA analysis is a true game-changer. As these lab-in-a-suitcase technologies become cheaper and more user-friendly, they will create a much more formidable deterrent for traffickers. The power of eDNA will continue to grow, revolutionizing how we monitor and manage shark populations, perhaps even through citizen science programs where everyday oceangoers help collect data. And as population genomics becomes more refined, it will provide an unprecedented ability to track the origins of the trade. But technology isn't a silver bullet. This global problem requires a global response, stronger international collaboration, more investment in training and capacity building, and rigorous enforcement of agreements like CITES. Ultimately, the demand for shark products drives this trade. This is where public awareness becomes a powerful tool for change. Studies that use DNA to expose mislabeling are critical for informing the public and pressuring the seafood industry to be more transparent. Calls for mandatory species-specific labeling for all seafood are growing louder. Right now in the US, sellers only have to label shark meat as shark. Stricter labeling would empower consumers to make informed choices and avoid products from threatened species. The story of the shark trade is a sobering one, but it's also a story of innovation and dedication. The DNA detectives on the front lines are providing the hard evidence needed to expose the truth and enforce the law. The tide has not yet turned, but with the continued advancement of science and the power of an informed public, there is hope that we can unmask the criminals, protect the vulnerable, and ensure sharks continue to grace our oceans for generations to come. Thank you for listening to Fun Facts Online.